of craziness. What you need to remember about this chapter is not everything. Everything. We want to think of this chapter, central nervous system, and this beginning, this chapter totally. This is the place where all information is either going to come to go to be processed, or to be sent away and to cause an effect. And what did we talk about when we talked about information coming in, information going out? How do we refer to that? Afferent, efferent, sensory, motor, information coming in, information going out. So the central nervous system consists of what? The brain and the spinal cord. And then the term cephalization is something that is a term that describes how the central nervous system in humans has evolved. So development of a front portion of the central nervous system, an increase in all of those cells that are going to send messages to and from the central nervous system to the head, and then the highest level reached in the human brain. So cephalad refers to what? The evolution of that top portion of the brain. So kind of ironic that it's at the top of everything in your body. So as we developed embryonically, and I'm looking at page 429 in your textbook, we see a little diagram there, figure 12.1. Yes, hon? Oh, you can't hear me? Wow. I've never had to use the microphone before. Yeah, maybe you need to move up. I'm pretty loud already. So, when we look at this diagram, we see embryonic development as the brain develops, and the central nervous system develops from embryonic tissue. When we talk about embryology, we see that you come from one cell. Did you know that? And that one cell starts to multiply, and as it multiplies into many, many cells, it starts to separate into different layers of tissue. So when we talk about the brain and the spinal cord and the central nervous system, we're talking about tissue that forms something called a neural tube. That neural tube will eventually become all the different parts of your central nervous system. So, 3 primary vessels form at the anterior end, the top of this tube that keeps twisting and twisting itself during embryonic development. So, forebrain, front, prosencephalon, mesencephalon, midbrain, and rhodoencephalon, hindbrain. So, the back part, the posterior end, the bottom part of this tube will eventually become what? The spinal cord. So, embryonic development, just a general overview, general terms from figure 12.1 is what I want you to know. So, we end up with a front brain, a midbrain, and a hindbrain as the embryo develops. 5 secondary brain vesicles, regions of the brain. And all these different regions, we're going to find out as we study this chapter, are different areas where different types of processing is going to take place. I keep referring to the brain as a bunch of file cabinets. But when we talk about different portions of the brain, we're going to talk about what's in those file cabinets. What gets processed there? Why do I have to access these regions for what type of information? So, again, when we look under developmental embryonic development, we see terms, basic terms that you should be able to recognize in the developing brain. We see that in your notes telencephalon, diencephalon, mesencephalon, metaencephalon. All of these are the developing brain, and they're eventually going to become, in the developed brain, when you are born, what's in the dark to the right. So we see the cerebral hemispheres. And we're going to talk about all of these different parts in this chapter. Epithalamus, thalamus, hypothalamus, the retina. What's the retina? Yeah, that's in your eyeball. Well, what does that have to do with the central nervous system? The retina. Isn't that like seeing? Don't I see with my eyes? Yeah, I don't see with my eyes. What do I see with? My brain. The information, the sensory information to process sight comes from the retina. But I see, I process, I visualize, I'm aware of sight with my brain. Everybody know what lobe? The occipital lobe of my brain. Okay, so we'll learn that that information coming in is extremely important, that sensory information. We'll discuss those particular sensory organs next semester when we begin. Next semester. Special senses. We also have a bunch of different areas in the brain that aren't necessarily file cabinets, but they're roads to the file cabinets. In order to get information from the file cabinets, what do I have to be able to do?
Access them.
Open the drawer and grab the file, yes? I have to access them. So a lot of these regions that we're going to discuss in the brain aren't necessarily file cabinets, groups of nuclei that process information, but they might be roads to the information. Does that make sense? So when we talk about things like midbrain and pons and the cerebellum and my favorite word in science, medulla oblongata. I don't know why I like that word, but I enjoy that word. And those are different places where I go to get information, where sensory information is rooted to those areas, the proper file cabinets to access information. For example, when information comes in from my eyes, it has to go get files from where? What lobe did we just say? The occipital lobe. Well, that's all the way back here. How the heck am I going to get from here to all the way back here? See where I'm going with that? So these roads or pathways are sort of like little hitches in a railway station that take information and make sure it gets to where it needs to go for processing. So as we go on, we'll talk about that as well. So everything on the right, that's the adult brain. Everything on the left, that's the embryonic brain as it develops. So, the brain grows faster than the skull. So it would be a good idea if we didn't have bones until later on, yes? Probably wouldn't work too very well if we developed our bones too early on in embryonic development. So we see that the brain, growing very quickly, continues to grow down through the brain stem. Brain stem leads to spinal cord. And most of the file cabinets can be found in places called the cerebral hemispheres. Why do I say hemispheres? It implies there's more than one, doesn't it? Well, there's actually two. There's a right side and there's a left side. So different file cabinets, different information is going to be processed in different places on different sides. Some of the information is processed on both sides, depending on what that information is. For example, your visual information from your two eyes is processed on both sides of your occipital lobe. Okay? So we'll talk about that as we go through these. So cerebral hemispheres are going to organize lots of information for us. Again, when you look at this diagram, general, think general. Is this the adult brain? No. What does it say? 5 weeks. 5 weeks of fetal development. You're already starting to develop your central nervous system. So we start to see that tube starting to do what? Twist and fold and turn on itself. And we start to develop these different areas. So colors in this are kind of nice because it shows you consistency throughout the embryonic brain. And as we go through, we see those same colors, 13 weeks, and then, where's my other one? There we go, at birth. So the colors stay consistent here. We can see this diagram on page 430 in your textbook on figure 12.2. So the adult brain regions that we're going to discuss in this chapter: cerebral hemispheres, my big file cabinets, the diencephalon, kind of central brain region that does a whole bunch of good things for me, the brain stem, which helps to feed information in and out of that processing area, midbrain pons and medulla, and then the cerebellum, which kind of hangs off the back end of the brain, kind of underneath those cerebral hemispheres. This guy is very important for coordination and organization of body movements. So you might have noticed, you probably, if you're observant at all, I tend to talk quite a bit with my hands. Yes? It's because I'm Italian. It's a curse. What am I going to do? But movement of stuff like that is coordinated by the cerebellum, processing. Me moving my hands, my feet, moving through space. We're going to see the cerebellum is highly involved in coordinated movements, skeletal muscle movements. So here we see the adult brain. So that tube keeps folding and folding and folding on itself until we see the highly folded cerebral hemispheres. The colors remain consistent again, and we see that purple region ends up being the diencephalon of the central, and then the cerebellum at the bottom, and the brain stem consisting of those 3 regions. Again, those 3 regions are very important. We call them lower brain functions, but they're still things that we can't survive without, even though we call them lower brain functions. So things like breathing and our heart rate are constantly being controlled by some of those lower brain regions. So, spinal cord, what's it for? Receive and send messages throughout the body. So it's a kind of connection to most of all the nervous system outside the central nervous system. What was that called? The peripheral nervous system. Okay? So spinal cord, extremely important. Pathways. There is some processing done in the spinal cord as well, and some information that's going to help be stored. Remember those reflexes we talked about at the end of class last period? I won't drop a book or throw anything at you, so don't worry. Remember those things? Well, there are some nuclei inside the spinal cord as well. So when we look at the spinal cord, we see 2 types of tissue. We see gray matter and we see white matter. Now, I think I explained this last class. Does anybody remember what the difference is? White matter is what? Myelinated fibers. So when we're talking about those myelinated axons, remember the myelin sheath? That's my insulation, right? So a lot of white matter, when we talk about white matter in the central nervous system, these are the roads I keep talking about. The pathways going to and from. There's different groups that will go to different places. But this is kind of the switching station, the spinal cord. This is where things are going to get highly organized and told exactly where to go for processing once they hit the top, the brain. And then the gray regions are what? The unmyelinated fibers and some of those nuclei. So the brain itself has a similar pattern. So if we look at tissue when we dissect the brain, it's going to be a little while from now because we have to finish all that bone stuff in lab. But when we hit the brain, we're going to look at a sheep brain. And we're going to see that there are distinct regions in the brain, gray matter and white matter. Same thing. The gray matter is what? When we're talking about the cerebral cortex, gray matter and the cerebral hemispheres, those are the file cabinets. This is where the nuclei are. This is where the processing takes place. The outer gray matter is called the cerebral cortex. And then we'll see scattered regions of gray matter as we come through into the brain stem. So a lot of the brain stem, central brain regions, we still have gray matter in there. Processing takes place. But a lot of it's the switching station, the white matter, sending things to where they need to go. Now, in order to make sure all the cells, the many, many cells of the central nervous system are fed properly, they have to be bathed in a ultrafiltrate of plasma, which means which cells make this stuff? We learned that in chapter 11. Nope. We're in the brain, no bone marrow. There's a group of glial cells that made this fluid that we're going to talk about. First of all, what's the fluid? And the cell's name is right up there on the screen in black. What's the name of those cells? Ependymal cells. Very good. Those are glial cells that make this wonderful fluid. And what's the fluid called?
The first sentence.
Very good. Cerebrospinal fluid. That's an ultrafiltrate of plasma. It's very highly specific because the ependymal cells make it very highly specific. And that is going to bathe the entire central nervous system, brain and spinal cord. So we're going to find some spaces within the brain that run down a little chute, kind of like a water slide that runs through the central portion of the spinal cord. So we have ventricles, brain ventricles. How many ventricles do we have? 4. Very good. So we have 2 lateral ventricles. Ooh. Lateral. What's that mean? On the sides. And I'm referring, if you look at them, you can see them nicely outlined for you on page 431, figure 12.3. So we have 2 lateral ventricles that feed into something called the third ventricle via the what? No, third ventricle. Laterals to third. The interventricular foramen. The third ventricle is then going to drain down into the fourth ventricle via the cerebral aqueduct. You with me? Again, on page 431, figure 12.3, we see this diagram showing us those ventricles in blue. Those lateral ventricles in the cerebral hemispheres are separated by a wall or a septum pellucida. Third ventricles in the diencephalon, central brain region. Fourth ventricles in the hind brain. What's a hind brain? Yeah, in the behind and lower portion. So the blue portions of this diagram is where we see our brain ventricles. Yes? Remember all those folds and grooves that were created as the tube kept folding on itself? Those just aren't haphazard. They're actually highly organized folds and grooves. So when I look at the surface of the brain, I see all these little ridges and folds. Some of them are just bumps, and it's kind of like our roads in Maine. And some of them will go way down deep. And you know when you think you're going over a small bump on the road and you go way down deep, it probably involves a trip to some sort of auto dealer to deal with your tires, yes? We'll see the same thing regarding the folds in the brain. So we have surface markings, and this is referring to the diagram on page 432, figure 12.4. So we see gyri. What's a gyri? A bump in the road. Shallow grooves. Sulci. What's a sulci? You're getting a little bit deeper. And then that bump in the road when you have to go visit the car dealer. I refer to those as fissures because they go way down deep. And sometimes they can actually separate different brain regions. So these bone markings, these brain markings, surface markings, can actually delineate different parts of the brain for us. There's a huge fissure that sort of separates the two hemispheres called the longitudinal fissure. And then there's transverse fissure, which is another big one, that separates that cerebral hemisphere from the cerebellum. When you dissect the brain, it'll make much more sense. And you can see them on this picture on page 432. So when we talk about the brain, there's different regions. And boy, should those terms look familiar. Why do those terms look familiar? Except for the last one. So points to your frontal lobe. There you go. How'd you know it was there? You haven't even started the brain yet. Because it's named for what? The bone in which it lies underneath. So put your hands on your parietal lobes. Yes. Temporal lobes. Yes. Occipital. Behind. Insula. When something is insulated, what does it tend to have associated with it? There's something covering around it, yes? So where might be the insula region? Inside, yes? So these brain regions all have different functions. You should understand and be familiar with the cerebral hemispheres. Different brain folds, or what we saw in the previous diagram, are going to delineate some of these different areas. So the next portion of your notes describes some of these areas that have been delineated. The central sulcus is an area that's going to separate parietal lobes from frontal lobes. The parieto-occipital sulcus is going to separate what? Yeah, so it's right in the word. So if you remember what the lobes are and you see the word, yes, then you can understand where they are located in the brain. So central sulcus, parieto-occipital sulcus, lateral sulcus, three basic brain regions, cerebral cortex, we have white matter, and we have the basal nuclei. And the basal nuclei is kind of that internal stuff. White matter, what are we going to do with that? What's it for? Yeah, those are pathways or roadways. Cerebral cortex, file cabinets, right? This is where we're going to store information. About 2 to 4 millimeters thin, our cerebral cortex is. Superficial layer of gray matter. 40% of your brain mass. And this is where the file cabinets are. I'm just going through these because these are some of the, you can see some of those lines that were described in your notes. You can see how it delineates some of the different areas of the brain. You can see that, what's that big, huge, long, cavernous fissure that separates the two hemispheres? Longitudinal fissure. And then we see the transverse fissure here, separating the cerebral cortex from the, what's that little, kind of looks like a little brain almost. The cerebellum. What's all those dark things? Yeah, those are blood vessels. This unfortunately is no longer living. So what happens to the blood instead of blood vessel in a no longer living thing? Yeah, it stops and then it dies and then it turns brown. So when you look at a specimen, when you look at your specimen, you'll see all those little blood vessels. All the little folds. Can you see them all? That used to be a tube, remember? Before it started to fold in on itself, it used to be a straight tube. So, gray matter, 40% of the brain mass. This is where conscious awareness, your files are stored. So, your perception, sensory perception. Just think awareness, motor, voluntary, communication. That's where ESP, extrasensory perception. We don't use a lot of the 40% of our brain mass. We don't use a lot of it. So is it possible that there are individuals amongst us that can use a lot more of their brain than we can, the average bear? Huh? Say,
I think you would be very limiting.
yeah, I agree with that. You know, who said, you know, we don't know so much. So how is it that we can, you know, say that these things don't exist when we can only use a certain portion of our brain? Maybe there are individuals that can use much more of their brain. Well, we didn't, you know, unfortunately, there's a lot of the science that we have now that can actually analyze that stuff wasn't around in his day, but who knows? I mean, if we threw him under a PET scan and looked at his brain activity, it might, you know, blow us away. But unfortunately, we didn't have that technology back then. So who knows? That's what I say. Keep an open mind. So, this is where all of the files are kept. Now, we don't want to just shove, has anybody ever been into an office where all the files are like all over the place? And the only person in the office that knows where it is the lady that's been there for 700 years because she made up the system? Yes? It's crazy, isn't it? It wouldn't make sense if we just shove files in all the drawers. We have to have some sort of organization to the files. Just like in your brain, there's some sort of organization to the files. So there's different areas where I'm going to file different things. Remember, I have 3 types of information, 3 types of hands-off going on in the central nervous system. I have information coming in. What's that called? Information coming in. Oh, come on, it's right up there. Well, it's not really, the word is up there, but you should recognize it's information coming in. Mm-hmm. Sensory information, information coming in. Then we have motor information, information going out. And there's got to be some kind of in-between place where I hand off after processing information that came in to information going out. What's that called? It's called an association area. We saw it referred to as an integration region in the spinal cord when we talked about what? You heard that word the end of last class. What was the integration neuron? Where was that? It's a good thing I got a large coffee today. It's mostly decaf. I did put a little pumpkin spice in there to add a little flavor, tastiness. Nobody? Integration neuron. We talked about some right at the end of class. And then you need to know these 5 things of a something or other. What's this? Where was it? What did it do? Yeah, what was it called again? No. What was that called again? Oh, thank you. I hate to do that.
You gotta do that.
Well, I said it before I was going to do it. I didn't promise I wasn't going to do it at all. So the association or interneuron was that handoff guide in the simple reflex arc. So in the brain, one of the functional areas is the handoff guide, the association area, where we're going to hand off information from sensory and motor processing. Okay? So each hemisphere has information, files, concerned with contralateral sides of body. And what the hell does that mean? Right, so when I go like this, who's taking care of that business? This. Yes? And when I go like this, who's taking care of that business? That. That's what that means. So you learned a big word. Why don't they just say the right side takes care of the left side? Oh. Scientists, they like big words. So, we see something called lateralization of cortical functions in the hemispheres. And that's basically what that means. Your conscious behavior involves all of these areas working together so that you can be aware of what's going on around you. We're getting so much stimulus all the time. It's overwhelming. And some people don't have a filter to all the stimulus that you're getting. I'll give you an example. You think you're just relaxing in your chair, enjoying an exciting lecture on the cerebral hemispheres, don't you? But that's not just what's going on. You have light information coming in. You have pressure from your clothes. Some of you have a ponytail in. Holy moly, all the hairs in your head are sending information to your brain regarding the stretch and the pull on your head. Your butt's sitting on the chair. All of those receptors are sending information to your brain. You're probably leaning on something, so information's coming from your elbows to your brain for processing. You are holding your head upright, so there's information being sent to and from, so the brain is telling you not to slump over, contracting certain muscles so you can sit up in your chair. You're also listening to me, so hopefully you're processing all the exciting information that's coming in from your ears and the receptors there. Holy crap! There you go. So you have temperature receptors that are firing off too.
The best place to get this lecture would be
In space. We don't have any of that pressure to deal with. So all of that information is constantly being sent to and from your central nervous system. So we have different areas that are going to help filter out conscious awareness of some of that stuff. You know, sometimes you'll be driving down the road. Has this ever happened to you? It happens to me pretty much every morning when I drive to work. I look around and go, oh, I'm here already. Did I fall asleep the whole time when I was driving? Hopefully not. So we're not always aware of every single thing that's coming in, was my point of that long, drawn-out, scary explanation of what was being sent in. So, cortical function is lateralized into the separate hemispheres. Conscious behavior is going to involve the entire cortex, and there is some filtering of information as well. This is functional neuroimaging, fMRI. What's an MRI? Magnetic resonance imaging of the cerebral cortex. And you can see all these different colors going on. And different colors are delineated by sometimes heat production in some of these different tests. So, speaking and hearing increase activity, blood flow shown in yellow, in orange areas, posterior, frontal, and superior and temporal lobes respectively. So different things going on, different processing going on, will give off different heat signals. And we can see them as different colors. You see these areas active in speech and hearing in this MRI. Excuse my little clicker thing. But they'll light up as brain is active in different areas in some of these tests. This is the thing that would have been nice to have if, you know, to look at brains of Albert Einstein and all these super smart people. Thomas Edison maybe would have been exciting to have a look at, to see if they have different function than the normal brain. So yes, it's clicker time.
Is that like hammer time?
Not quite. I refuse to dance, and I don't have big hands. Here, touch this. All right. So, which of the following best explains the convolutions and folds present in an adult brain?
Is that like A, B, and C?
Yeah, that's A, B, and C. Oops. Sorry. Start again. That's all we have, 23? 5. Oh, there we go. That's better. 4, 3, 2. Did everybody click? We have 45. Good. What's the answer? Shall I? Oh, there's a D. No? Okay. The answer, wait a minute. The answer is C. Get? Yes? Because if we didn't have all those folds, but we still needed all those file cabinets, yes? Motor areas of the cerebral, oh, wait a minute, that's not a question. That's not a question. Stop. All right, get small now. Display, yeah, nobody got it right, I know. Okay. Oh, stop it. Okay, so what I want you now to do as you go through your notes is I want you to understand where all these file cabinets are and what their purpose is. So first we're going to talk about motor areas. And when you look in your textbook, it starts on page 433, described under motor areas. What I've given you in the notes is what I want you to know. Okay? So we're going to start out with motor area. Motor area is where? Yeah, in the frontal lobe. What's it control? Voluntary movement. Motor, movement, voluntary movement. Is there involuntary movement? Yes. Where is it? Give me an example. Your heartbeat, your breathing, all of the muscles that need to contract in your digestive system to help your breakfast go through. Yes? Blinking's not involuntary. No, you do it sometimes unconsciously, but see? So that there's a difference. Yes? Sometimes we do things unconsciously that are still categorized as voluntary. You know, like me driving to work. Well, if I was 14 years old, the drive wouldn't be so easy, right? How come? Because I wouldn't know how to drive the car yet. Well, sometimes they learn early. But, you know what I'm saying? Or play the piano, or the type. Those aren't unconscious. Or even though we sometimes think they are. We're sometimes unaware because we've done that so many times that it comes naturally to us to do that. Therefore, if we want something to come naturally to us without effort, what must we do? How did I get good at driving? I did it a lot. Although my husband still says I'm not good at it, but, you know, he's a guy. I'm good at typing. Why am I good at typing? Because I do it a lot. You see where I'm going with that? So wherever those files are stored, that road is very well worn to get that information to perform that function. Whatever it is you want to come easy doesn't come easy in the beginning. What do we have to do? Repetition, 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 repetition. Even when we study for our anatomy. Yes? Because believe it or not, when I say this stuff and you read it once, you've made a file for it. It's in there. It's in the cabinet. Now why can't I just go get it? Go get it. Yeah, no pathway. You have to make the pathway. You can't just walk it once. It's like walking through the woods. Yes? Anybody? Well, you guys are Mainers, yes? You know, walking through the woods. Well, sometimes you walk through the woods and it's like, oh, there's a path there. Why is there a path there? Because somebody's walking it a lot. Whether it be a moose or a deer or you might go in there and clear a path. You see where I'm going with this? Okay, good. So, primary somatic motor cortex in the precentral gyrus. Premotor cortex, anterior to precentral gyrus, are different motor areas. Again, they're outlined in your outline. Have a general idea of what they do. Broca's area. We're going to talk about what they do in a minute. This is just saying where they are. Anterior to inferior premotor area. You don't have to remember it like that. Just be able to point to it. Look at some of these diagrams. The one that's on page 434 shows us where these areas are. And then we have the frontal eye region. What's a frontal eye region for? Seeing things in front of us. What lobe is it in? The frontal, what do I, oh, okay. I process vision in the occipital lobe. But how do I get it there? How do I get the information to the occipital lobe for processing? Through what?
Nerves attached to you.
Through these guys, right? I have two of them, and I have to move them around to get information in for vision, right? Those are the areas. To move my guys around so I can get the information in for processing. So that's what the frontal eye field controls. Things like your eye muscles, muscles inside your eye called your iris that are going to control the amount of light that comes into your eye. That's what that's talking about. So this diagram here on page 434 shows us some of these areas, where they are in the brain. Again, we're talking primary motor cortex. So we're talking about information that does what? Motor is what? Movement. So, allows for conscious control of precise skeletal muscle movements. Innervates different body regions. The right arm, the right leg, the left leg. This is kind of a cool diagram on page 435 because it shows or outlines some of the different motor maps and sensory maps in those regions. So primary motor cortex is there, and those different areas control those different parts of the body. So the toes, the genitals in this region, the knee, the hand in this region, the neck, the brow. You see where we're going? Isn't that a cool diagram? I like it. This is one of the coolest diagrams.
It's a good thing we don't look like that, though.
Yeah. But it shows those different regions where the files are kept for those different parts of the body. Same thing here with sensory. So sensory map for those different parts of the body. They look kind of the same, don't they? And who do you think hands off information in between these two guys? So I poke you in the eyes, that's sensory information. How do I know how to blink? Motor ear, and the integration areas are going to kind of do what? Connect the two. So the premotor cortex, what is that? Well, that's going to help with planning out movements. Skilled motor activities. Believe it or not, walking is a skilled motor activity. And sometimes I have no skill. But I have to plan it out. Even though I'm standing here and then I would just walk over here like this, kind of unaware of that, I wasn't always able to do that, was I? When I was little, I had to learn that. I had to make files in those regions for those actions. Because I've done them so many times, they come seamlessly. But those are all things that I've had to create files for. Controls learned, repetitious patterns, motor skills, a lot of the things I've been talking about today. When damage to these areas occurs, that's when we are so very aware of them. When damage to these areas occur and we want to go someplace to get something, but we can't. When motor areas are damaged and sensory areas aren't, we can see it so evident because we might have a patient, for example, that has a stroke.
My mom felt one that had a few weeks ago, got a brain bleed.
Mm-hmm.
She can't read anymore. She can still write.
She can write, but she can't read.
She can see the letters, but she can't. She said it looks like it's written in Russian.
Mhm.
Or something.
Because there's an area in her brain that was damaged or temporarily, hopefully temporarily, disrupted, that puts those letters together for us. So we can see that when we see damage to those areas, how important those files become. So, skilled movements, premotor cortex. Voluntary actions that depend on sensory feedback. When I walk across the stage, it's a flat area. But when I get to the end, what happens? Gosh, there's enough chairs here. Let me do it here.
It becomes a drop.
How do I know that? Can I just keep walking like I walked across the flat area and go down
Your
there?
eyes see it.
No, I will follow my, yes? My eyes are giving me information about what I have to do. What do I have to do? I can't just do this anymore. I have to do this. Yes? Different movement, different muscles, different message to those groups of muscles to create that different movement. Where did I get that information from? Sensory. Yes? So those are the guys that are going to help coordinate the movements based on sensory information, like vision.
So what do you use when you walk around your house? That's completely different. You don't have to run inside.
No, but what you use some of your other senses of information. Now, you know, because of repetitive movement, like when I had to get up and go to the bathroom in the middle of the night, right? Unless the dog is laying there, I know it's a clear path. Son of a gun, doesn't she always want to lay there, right? So I know, because I know my room, I access those file cabinets. I see, okay, there's a clear path to the bathroom, because I've been in my room so many
You know
times.
exactly where you're going.
Yeah, and what I do, I don't know exactly where it is. I have to kind of go like this, right? And then sometimes I leave the door open, and I'm like, oh, left the door open. That's good, right? And then I always have to check to make sure the toilet seat's down, because I do live with my husband, God forbid, right? And I kind of know where that is, right? Based on all that information that I have in my files. So it just, again, it doesn't come naturally. It's stuff that you have in your files that you know to go access at that time. So somebody who doesn't have visual cues is going to do what? Rely on all the other cues about that information. So when you lose your sight, you realize it's not just your sight. You know, there's so many steps to the end of this stage. And I don't pay attention to that because I have my visual cues to help me. But someone who's lost that will then pay more attention to the other cues that they get as well. Broca's area. What's Broca's area for? Excuse me. And this present in only one hemisphere, usually the left. Usually the left. Do we all have the same brain map?
No.
Absolutely not. These are kind of the average of where they might be. But is there someone sitting in this room right now that has Broca's region in the right side of their brain? Yep, probably. So our brain maps are all different. Can you see what the neurologist goes through? When a patient comes in with a problem, it's not just an easy answer. And patients sometimes get frustrated when they say, oh, he's a doctor, for Christ's sake, he should know this. And we're not all wired the same. Some of us don't even have organs in the same place. They're a little bit lower, they're a little bit higher. Some of them are on the totally opposite side. Some of us have extra stuff. Yes? So Broca's region, typically on the left side, is for what? Speech. That's me talking to you. Now remember, speech just doesn't come. What else is involved in speech? You need to access files from other things, like sensory information that you heard. When I talk to you, I'm not screaming at the top of my lungs all the time because I know that I can hear myself speak at a certain loudness. So all of that information is being processed in Broca. Planning speech, voluntary motor activities, moving my mouth, breathing out so that I can create sound through my vocal folds. All of that's Broca's responsibility. And again, refer to this diagram. It shows you where everybody is. Where's Broca? Is it on this diagram? Yep. Yeah, see it, and where's, well, here's how I remember it. Where's your ears? Yes? So here, in this region. Yes? Question?
My husband says sometimes that I guess I really don't realize that I'm talking so loudly. And
Yeah, they say that to me too.
well, because of my car accident, I have busted my window out with this part of my head.
But it might not be your brain, it might just be your ears. Maybe, unfortunately, as we get older, our hearing starts to get a little off. So sometimes I will do the same thing.
I wonder if it's a certain type of hearing though, because in another class that I have, I sit at the front, but I can always hear these three young ladies at the back of the room who go back and forth. Even if it's not loud, I can still hear what they're saying.
Yeah.
It drives me half crazy.
Yeah, and different tones and different pitches we tend to cue in on too. So it has to do with our surroundings and what we're used to. One of the things that happens to us as we age too is we might also lose certain ranges of our hearing as well. I always use my husband as an example because he worked on submarines with engines, very high-pitched engines. He can't hear that anymore. He's lost the ability to hear that range. Unfortunately, it happens to be the same range in which the dryer alarm goes off. So I don't know if that's convenient or... But yeah, sometimes we'll lose different ranges. You know, you go to that rock concert way too many times, and you're gonna kill receptors in hearing receptors. And then sometimes we'll just start off, if we get excited, and be loud.
So I watched the show the other day that some shopping malls have, they play a frequency that only kids can hear.
Well, they have you might have heard this, and my daughter freaked me out the other day because it made me feel very old. Something called, it was a tone on her phone.
Yeah.
Mosquito. What did she call it? Mosquito tone. Did you guys know?
Supposedly only little kids.
Yeah, younger kids can hear it. So she played it on her phone. She's like, "Do you hear that?" And I'm like, "No."
That's my dad too.
Yeah, and she's like, "I hear it." It's like, So as we age, we lose some of our receptors, some of our information is lost. Sensory areas. So, not to forget the frontal eye field. What's the frontal eye field? Control voluntary eye movements. Remember, we need to move our eyes so we can collect that information for vision. So now we're into what? Those were motor areas, yes? What are we talking about now? Sensory areas. They start on page 435 in your textbook. See, this isn't as bad as you thought it was going to be, is it?
Worse.
No, it's not. Stop it. Glass half full, glass half full. So, this is conscious awareness of sensations. Somebody touching you. Light being processed. Sound waves being processed. Temperature being processed. Occurs where? Parietal lobes, insular, that inner region, temporal, occipital lobes. Some of the different areas where all of this processing is taking place. Primary somatosensory cortex, the somatosensory association cortex, that's kind of the in-between guy. We've got visual areas, we've got auditory areas, We've got something called a vestibular cortex, an olfactory cortex, a gustatory cortex. What the hell are those? Well, vestibular is for what? Balance. Olfactory, smell. Gustatory, taste. So all of those different areas process that information. Those things attract us to things that are going to help maintain homeostasis. Usually when I leave this classroom, my olfactory cortex is stimulated. As I walk from the Kirk building back to the Jalbert building, past the little area there, the cafeteria vents all of its cheeseburger smells. Yes? Why is that important? Cheeseburgers aren't really the best choice to help me with this, but yeah, they attract me to what? Food. Food is kind of important. Why is food important? Yeah, I have to go eat. I have to go seek out food. Unfortunately, I can't stick my feet in the ground and suck all the nutrients I need from it, so I have to go find it as a human. So all of this information is helping me to maintain homeostasis as a human. Visceral sensory. That's the thing you talked about when you said, who said you were a little cold and you probably should have worn pants. Yes? Why is that important to us as humans? Yeah, so we don't go outside in, 40 below weather in our bikinis. Not a good idea, right? So those receptors are sending us information about the environment around us. It's cold, it's hot. We're warm-blooded organisms, so we have to maintain a core body temperature. So those receptors are very important. I'll tell you a story. I got a new toy. Guess what it is? You know what it is. Hot tub! Woo! Can I hang out in there for like 3 or 4 hours? No. Even though I would like to do that, I can't. Why can't I? My visceral receptors will say, get out and get out now. They'll make, so I sat in it, first time I sat in it, quite nice, I might say. But you can't sit in there for more than 15, 20 minutes without doing what? Well, you don't really sweat because you're in the water. You don't really know. You start to feel real nauseous. My visceral receptors are saying to me, you're starting to get what? Too hot. You need to do something. They'll cause me to say, oh, I gotta get out of here. Otherwise, I'm gonna get too hot. Do you see what I mean? Those receptors are responsible for maintaining your homeostasis and getting you out of the hot tub before you really want to.
Some people don't want it. I've had one for 10 years and I sit in there for a while.
Well, what's your temperature at? Is it really? Oh, God. I
don't say that.
Different people have different tolerance. Yes, absolutely. You'll see different groups of people who are able to tolerate cold for a lot longer than others. Yeah. See, I the hormone heat's already there, so 15 minutes in, I'm like, oh. So, yeah, different people have different tolerances to different things. All right, so we're on the sensory side. Notice this. Where this stuff is. It's in a band in that central region, post-central gyrus, and some of the sensory map. Somatosensory cortex. Some of the different areas we need to know that are going to help us with some of the different information that we're receiving. Parietal lobe, the post-central gyri of the parietal lobe. Again, that stripe is what they're referring to. Here we're going to find information processing, receiving information from things like skin, proprioceptors. What the heck is a proprioceptor? You find them in skeletal muscle, joints, and tendons. What's going on there? First of all, I'm standing up. Why am I able to stand up? I'm getting information about me putting pressure where? On my feet. I'm getting information from receptors in my feet that there's weight there. Not only from my feet, but from when I do this, from who? My knees. When my knees bend like that, I'm sending information to my brain that tells me, okay, if she's going to bend her knees, we've got to do something with those muscles up in the top of her legs and something with those muscles down in the lower part of her leg region, right? That's information, again, coming from, that basically is telling us what's going on spatially with our bodies. You know, when I lean forward, different muscles have to contract so I don't fall on my face, right? When I lean back, different muscles have to contract so I don't fall back. Those are called proprioceptors, and they're going to help me with spatial discrimination, where I am in space, what muscles have to be contracted, what muscles have to be relaxed in order for different movements to take place. So again, we find those guys in skeletal muscle, in joints, in tendons. Also spatial discrimination. Have you ever come upon something and you feel like you're out of balance? Some people have problems with heights. They do this. Or one of the things that used to freak me out when I was a little kid was a fire escape. Because of the way the lines are in the stairs, I was it's not bad now, but when I was little, I remember this, being freaked out about walking up the stairs of a fire escape because I was thrown off spatially. Different cues from visual cues that are going to help with spatial discrimination are helped in this area. So, size, texture, those are all important. When I go to pick something up, if it's really, really big, have you ever seen those fake rocks on sets, movie sets? You'd assume that when you went over to the rock, that it was really, really big and really, really heavy. And if you're going to go pick it up, those cues would tell you to contract certain muscles to pick it up. Better example, my son, when he used to live at home, and the milk bottle in the refrigerator. I've opened the refrigerator and taken out the milk bottle many times in my life, so I already have a predetermined notion of how much I have to pick up, how many muscles I have to contract, and you know where I'm going with this, and to pick it up. But after my son has been around for a while, I go to pick up the milk and I smash it to the ceiling of the thing because the freaking thing's empty. Or there's like 1/2 of an ounce. It's like you couldn't finish that. It was just too much for you to hold it.
Why do they do that?
Oh, to drive you crazy. That's why they do it. That's their job. And he did it well when he was living at home. So you know where I'm going with that, right? Because all the cues from all the files that I'd had for that gallon of milk to lift it told me I had to contract my muscles a certain way and apply a certain force. Yes? That's what these guys are responsible for. So, primary visual is going to help me with that as well. Again, visual information is big. We get a lot of information from visual sense. And when we lose our visual sense, it takes a long time for us to really cue into some of the other keys of information we get. But the visual areas, primary visual cortex, is going to help with so many different things as far as movement goes through space. Posterior tip of the occipital lobe receives visual information from the retinas. So this is where we process that information. Visual association areas. Around that primary visual cortex. Again, interpret stimuli. This is where we have files for things like color, movement, the form of something. You know, I sit there and I look at this thing in front of me, and I know, because of all the files that I've stored away for so many years, that that's a chair. And I know that that chair is like a maroon color. Yes? How do I know that? Because the information that's being sent in visually is accessing files that know that that's a chair and what color it is. So visual association areas are going to help associate objects with what I see. Yes?
We're kind of, this is correlating with my critical thinking class. We're talking about now how, like when you buy a car and you drive it around and all of a sudden it's like, oh my God, they have a car like mine, they have a car like mine. It's not really, it's just that now that you have that, the pathways in your brain, now it's because you've seen it.
Mm-hmm.
So now you see it again and again and again. Nothing's really changed, it's just now that you've seen it, now you're seeing it everywhere.
You see it everywhere. You always saw it everywhere.
Right,
but now you're what?
Aware,
You're using
aware.
the word, you're more aware
Exactly.
of it. Exactly.
Okay.
So again, all this information is coming in all of the time. And we filter out quite a bit of it. Because it would be completely overwhelming to us to do that. Does anybody know anybody or any small children with different forms of autism? Okay, what's one of in some forms, there's so many different forms, but what's one of the things you have to be very careful with a child? Overstimulation, because they have a problem with the filter. When you send it, and it could be anything from touch or that ponytail in the hair. Some of these things can really overwhelm a patient with this disorder because they have a problem filtering out all of this stimulus. It's overwhelming to them. So it's important to be aware of that. All right, where are we? Auditory areas. And hopefully you understand what an auditory area is. Getting information from where? The ears. So temporal lobes interprets information from the inner ear, pitch, loudness, location. You can tell where sound is coming from. All of that's stored in your association area, primary association, auditory association area and primary auditory cortex. Vestibular cortex, what's that? Now that's that whole spatial thing, so I don't fall over. And there's tons and tons of information coming in as far as where you are in space, where you're standing. Do you have to get up? Do you have to sit down? Do you have to lay down? All of those things take a lot of processing because they take different muscle groups to contract to get you to where you're going. Anybody ever have vertigo or some kind of balance issues? Yeah, so you have a higher appreciation for all of that information coming in because you know how it can truly mess you up when that information gets messed up. You have problems walking, when you go from standing to sitting, when you go from sitting to lying. Huge problems if you have problems processing that information. And again, we'll find those in the vestibular cortex. The olfactory cortex. What are we talking about here? Smell. So the olfactory cortex, located where? Temporal lobe region. Olfactory bulbs, when we discuss the cranial nerves, we're going to talk about the first cranial nerve, and you're going to have to know all 12 of them and what they do for a living. But this cranial nerve, your first cranial nerve, is the olfactory nerve, and that is going to help send information for processing in the brain with respect to smell. Now, why is that important? Yeah, there's a fire. If you go to the refrigerator and you open up that little plastic container with the unidentified object left over in it, and a very bad smell emanates from it, you know not to do what with it? Don't eat it. It's what? It's bad. Yes? So very important information. Again, to help maintain homeostasis. Different smells can draw us to different things. We have a very low appreciation for our olfactory abilities because our olfactory bulbs are kind of dinky. Other animals use their smell for so much more. Like your dog, for example. That's how they're attracted to different areas because of smell. They know where they are because of smell. They know what's friendly, what's foe because of smell. Because it's so much more highly developed than we are. It's why when you take your dog out to a different place that they haven't been in a long, long time or they've never been to, what's one of the things your dog does? Pees on everything. Why? That's like leaving breadcrumbs. Because then they'll know how to get back. Because they'll be able to smell it. Plus sometimes, especially male dogs, will pee over that. To say, nope, mine. Yes? So we'll pick up with this on Tuesday. Listen, this is a crazy hard chapter. So take my advice. Start studying now. Okay? I hope so. Maybe. We'll try to. I want to ease into it. I'm going to speed up a tad. Make sure you sign the sign-in